What are you laughing at?
I'm laughing. Let me just say hello to everyone. Hello, everybody. Welcome to Q&A with A&V. I'm Vincent Racaniello, and joining me from New York, Amy Rosenfeld.
Hello. So, how was your day?
I'm laughing because we already have a thumbs down and we didn't even start. How was my day? So today, update on the incubator. For those of you who don't know, the incubator is our new studio that I'm sharing with Daniel Griffin. And I went today there directly from home and worked there for about 4 hours. I have some pictures. Should I show?
Yeah, we should show.
What I did there. So I built this little cabinet for tools and wires and parts and stuff that you need in a studio. It took me like an hour and a half to build it. And I don't have any chairs to sit on, so that was kind of pain. Wait, here we go. Then this is my communications closet where the internet comes in on this wire here, the single wire, and then it goes into the router, which is this white thing which I put in, and then it goes from the router to the switch, and from the switch to the patch panel, and then it goes all out into the room with all these individual Ethernets. And I also have Wi-Fi. So that's exciting. The internet is working. And finally, I have a desk. I have a raising desk. And someone on this live stream told me to get a rise and lower desk, so I did, and I think it's a good idea. This is from fully.com. And so this will be a working desk where I do editing, but for now it's all I have, so I may end up shooting on it. Okay, so that's the update on the incubator. And then at about 2 p.m. I was summoned northward to change the liquid nitrogen.
Yeah, I can't afford to have my cells die.
Oh, one more thing before we forget.
Ah, yes, it was. Thank you.
This week was Amy's birthday. Everyone wish Amy a happy birthday.
Thank you very much. Is that my virus?
It's a cake virus, though.
It is a cake virus, yes.
Yeah, I found a cake virus for you, so I got it. And yeah, Amy's birthday was Monday, and
that's true. And we have to make a public announcement about your vaccine town hall tomorrow at 7 for ASV.
You have to sign up for that ahead of time, though, right?
I'm sure it's like 2 seconds ahead of time. But anybody who knows anybody who has questions about the vaccine or something, then they can go email you and talk to you tomorrow night at 7. So it's good.
And welcome, Frank and Vanity Nutrition, our moderators for tonight. We appreciate your help a lot.
Yeah.
Be out of control without you, so really appreciate it. All right, let's get down to the questions. From Mike, this guy at Oxford said herd immunity is not possible as Delta infects the vaccinated. How is he wrong? Amy.
What, Amy? I'm on vacation. It's my birthday.
Okay.
No, I'm just teasing. What? Amy, what?
Why is he wrong? Somebody's singing you happy birthday.
That's cute. That's very nice. Thank you. Well, first of all, Delta is SARS-CoV-2. So if Delta is infecting vaccinated, other variants or isolates are infecting infecting the vaccinated. Number one. And number two, it's not clear to me that it's transmitting between vaccinated and unvaccinated people. So, I mean, so if it's not transmitting, I don't know why you wouldn't be able to get herd immunity.
Yes.
So here's the problem is like everybody has different assays and everybody's taking single time points and stuff. And so you have the Chinese group saying, it more rapidly reproduces and transmits or whatever. And then you have the Singapore group that says it's aborted. And then you have Dave Connor from Wisconsin coming out today and saying, Well, even at a low CT value in vaccinated people, it's still infectious virus. I did the infectious virus assay. So, I mean,
How much?
now we have an answer. We have one of every possible combination almost. So, who knows?
The bottom line, as Amy said, herd immunity depends on inhibiting transmission, not infection. And
Right.
so
And
you
it
get herd
doesn't seem.
immunity with polio vaccines, and they don't prevent infection. So, Dr. Pollard, go back to your textbooks, please, and read them and not just talk off the cuff.
Well, you get herd immunity in measles, and that, in fact, that vaccine doesn't
Yeah.
protect whatever.
Do you think Novavax is the best vaccine? Will it be better at resisting breakthrough as the spike protein doesn't have to be translated, transcribed by our cells, which introduces mutations?
No. So first of all, I think that he's a bit confused about the fundamental central principle of biology. So DNA to RNA is transcription. RNA to protein is translation. There's no mutations in that process of translation. Very few, very rare does that wrong amino acid get incorporated. Um, so it
Yeah.
doesn't, and it would only. It's not a mutation; it would be an alteration. And since the same mRNA is used to synthesize multiple copies of the protein, it would, even if it happened once, it would be no big deal.
Yeah.
And so Novavax has, I haven't seen any data on Novavax. And then I read this article recently, or I skimmed this article recently, that they're like even further behind.
Hmm. So, Eric, it could be good, but we don't know. And it's not an issue with mutation. So, as Amy said, no worries. Cardiologist Topol. Oops, did I say that? Making the waves again with nonsense says vaccines now below 40% effective, according to a Mayo Clinic preprint. Effective at what? Preventing infection. We don't care about infection.
Oh,
We
it's
care about
preventing
disease.
infection? I thought it was preventing, I don't know, sneezing. I don't know.
Topol doesn't get the difference between infection and disease, I suppose. But you
I would
notice
think as a cardiologist you should, because there are viruses that cause disease in cardiomyocytes and stuff. So
some of
I
your
would think,
viruses, right?
yeah, some
Some
of my viruses do. So I would think that he would get the difference between infection and disease, but I guess I was wrong.
Of course, everyone here from California knows that your lovely state is mandating vaccination of teachers and kids in the schools, and we applaud you. This is wonderful. Congratulations. I just heard about antibody-mediated enhancement. What is it? Why is it not a concern with vaccines and the vaccinated? Yeah, we talked about this last time, narcissisectomy, and you were here, I think. You want to do it briefly, Amy? Very briefly. ADE?
So there is no ADE in
And COVID,
people.
right?
Yeah, there is no ADE in COVID. Um, so this is where animal models were not exactly truthful. Like people thought they saw ADE for SARS-CoV-1, but in mice, but you never saw it in people. And so ADE is where the antibody that bound to the virus then binds to like an FC receptor and it facilitates its increased uptake. Right? Is that
Yeah,
right?
yeah.
Yeah.
And I showed a slide
With
last week.
dengue, it's usually
Yeah,
it's usually talked about in terms of dengue.
right.
Um, but yeah, it's not. Yeah.
Not a concern with COVID vaccines. Narcissist tech. I can't even
Uh,
pronounce it. Jennifer, just read that Stanford is mandating weekly COVID tests. I hope antigen, regardless of vaccination status. Do we really know what positive test, especially PCR, means in a vaccinated person?
no, that's why I'm not a big advocate for testing vaccinated people.
Yeah, we don't know what that means. And there, as Amy pointed out above, there's evidence out of Singapore that your copy number drops very quickly in a vaccinated person compared to unvaccinated. So, no, we don't know what it means. And for the CDC to have said last week that it means people are being infected and they're transmitting is not correct, because we don't have the data. She goes on. I remember Drosten in German said Germany used lower cycle CT values to represent positive cases, which explained fewer asymptomatic cases in Germany.
I don't think that that's true because there's no correlation with infectious dose or load and symptoms. It's not HIV.
Right.
So I'm not clear that he's talking about. Are you?
Nope. No idea. My 4-year-old is starting school in September. We live in Ontario. I'm nervous. Any advice? Vaccination rates are fairly high in Ontario, but cases are going up. All right, so
Wear a
4-year-old.
mask.
Wear a mask. Yep. Absolutely. I think that's the key. If you want to send your child to school, wear a mask. And then this is following up on this, the school lunchtime where kids take their masks off, the most risky time of day. I would think so. What do you think, Amy?
Yeah. I mean, my aunt asked about her granddaughter who's not vaccinated. She's only 9, and they live in Florida, and the school is not expanding the lunch area, so the kids are all sitting on top of each other. It's not staggering times and stuff. And she's very upset about it. They have the girl promising she's going to take a bite and pull up her mask and chew. Take a bite, pull up her mask and chew. I'm like, okay, good luck to you.
Yeah, lunch is a tough time. Masks off, closeness, chatting. Yeah.
Yeah.
Taking the kid home might not be a bad idea.
Yeah, but not every school can do that.
No, of course. Jennifer, thank you for your support. We will use it for supporting the incubator, the site of good science communication. All right, Armin wants to know what defines the tropism of mRNA vaccines towards the antigen-presenting cells. What cells produce spike protein? Only APCs or any kind of somatic cells?
I
Right,
don't know
Amy?
yeah. I think it's any cell, any
pretty
APC or muscle cell. I don't think that there is any protein that targets the lipid droplet to a specific cell, like
Right.
a receptor.
Yeah, it goes into all cells. And, of course, in an APC, it's great because it can present it to a T cell. But it can also be sampled on the plasma membrane of a non-APC, a somatic cell.
Yeah.
What is the all-time best movie about a virus outbreak for any reason, and why? do you don't watch movies, do you, Amy?
Not really. I have an attention deficit.
Well, I think Contagion is probably the best. It's the most accurate, which is not to say it doesn't have mistakes. There are certainly mistakes, and for my virology course, I usually assign it as extra credit to watch the movie and write down 3 mistakes that they made in the movie, and everybody gets 3 mistakes easily. But the reason it's more accurate than the others is because our good colleague Ian Lipkin was the script consultant, and we collaborate with Ian. Amy sees Ian frequently, right?
Yeah, I see him at least. I see him. I see him minimally every 10 days, sometimes more. Depends on how often I go across the street.
So I think Contagion does it, but they're mistakes. And in fact, you guys can tell us what you think of the mistakes if you remember it, or else I'll ask you in another.
Well, they could
live stream.
rewatch it.
If you rewatch it, yeah. Is it possible that if many human cells are targeted by mRNA vaccine and produce spike, lack of co-stimulatory markers may lead to peripheral anergy? Because co-stimulatory markers, the co-stimulatory markers are on the T cells. And if on a somatic cell, it may be sampled by APCs, right? Dendritic cells could pull off some of the antigen and then bring it to a T cell. So I don't think that's an issue. But, Amy and I aren't immunologists. We're just virologists.
Gosh, this is outside the realm of my comfort level.
Do you know what cells express the mRNA, spike mRNA, and are tagged for attack by immune system? Can neurons, heart, egg cells express it too? I could not find any biodistribution studies to answer this. Well, there are. The companies have done biodistribution studies, and they are published, so they're out there. And what they do in a biodistribution study is they put a ton of mRNA in, a higher level than you will ever put in a person. And of course, it ends up in other places. But what I would say, Jamian, many millions of people have received these mRNA vaccines with very low frequency of untoward effects. So I don't think there's a worry there. What do you think, Amy?
I think it depends on how the animal was inoculated. Because if you put it in certain ways, it automatically goes to the liver because that's a filter organ, right?
Yeah, sure.
So I think I think you have to be very careful about what you say. And how much if you put in 1,000,000 times more, I don't know what it means.
Yes. And now Damien tells us there's this one study, increased mRNA were found in spleen and eye, could be detected in all tissue.
Spleen is a filter organ for immune cells.
Yeah, the lymphocytes will go there, so it's not surprising. But yeah, if you put enough in, you're going to find it in a lot of tissues. But I don't see that that's an issue, frankly, because we're putting very low levels in compared to these distribution studies. So I'm not too worried about that. And I have no stock in any of these companies, so I say it from a scientific viewpoint. I'm 70, male, going to social events, second Madeira 5 months ago. Some vaxxed olds getting COVID. Pfizer data shows third dose 100-fold increased antibodies. Why shouldn't I get a third? It's not necessary, right?
Right.
What was the paper that Daniel sent us recently? Was that right? What was the tweet he did? Well, he did tweet. It was a paper, and he said...
He tweeted, he said, where's the science data? So, yeah. So it's unclear to me when the data was taken to show this 100-fold increase of antibodies. So if I gave you a third dose and I came back, I don't know, 11 days later, of course I'm going to see a big bolus of antibodies, right? I just boosted you.
Yeah, right.
So I don't know what it means.
Yeah, I totally agree.
And then it's going to decay at a certain rate. And I bet you if I plot that line of decay after the spike, the spike, and then it starts to go down, I bet you that decay rate is the same after the second dose. So what have we done? We've done nothing. It's not necessary.
Increased antibodies doesn't tell you anything.
Not only that, it's unclear to me that these older people who are getting COVID are getting severe disease or not just a headache and some sniffles. You just came and said your friends have some or vaccinated and got infected and they have a headache and sniffles, which is the same as somebody else who's in his 20s.
Yeah, I think that some older people die because the vaccines are not 100%.
Well, nothing's 100%. But
yes.
Even if...
Only death.
Well, only death, yes. But even still, I bet you that percentage of older people that's dying after vaccinated from from an infection after being vaccinated is probably 1,000,000 times less.
Okay.
Don't you think?
Yeah, I agree. Let's see. Do the vaccines created spike proteins protect both host and virus from each other and thus allow a huge amount of virus to increasingly invade? If the protective protein fails, will we all perish? No. No. Doesn't do. The spike protein induces an immune response which protects us against the virus. It doesn't do anything for the virus. Right, Amy?
Right.
Uh-oh, Angie Rasmussen. Transmission
Okay.
has outcompeted evasion of neutralizing antibodies and variants. No, no, no. No evidence for that. I'm sorry. There's no evidence that transmission
I,
has outcompeted evasion of neutralizing antibodies. And it's not just antibodies that inhibit transmission. It's also cytotoxic T cells. Dr. Rasmussen is wrong. Sorry, Amy, do you want to correct me?
no, I'm just flabbergasted that somebody is quoting Angie to us. Sorry.
I have to show
I'm going
MK's
to need some booze.
little icon. I love the little sculpture there.
This is
Tea and
good.
knitting. That's good. There was a virologist who used to come to seminars. She was at Caltech, and she would sit in the front row. She and her husband were both professors at Caltech, both virologists, and she would knit in the front row.
Is that Strauss?
Yeah, Strauss, Mrs. Strauss. And I somehow found it very disconcerting. I don't know why.
Well, I would find it very disconcerting. Why aren't you listening to what I have to say?
Well, maybe she is, right?
I don't know. But all I know about them is that Charlie got his PhD with them.
Hello, Tom, from Western Oregon. So that's where my desk came from, somewhere in Oregon. It shipped all the way, made in the U.S. in Oregon. Very nicely made desk. Lovely. A lot of contagion stuff here. Yep. What would your most accurate guess be in regards to the current situation had no lockdowns occurred? More infections and more deaths, but still waves. As Jeff Shaman said on Twiv, which will drop tonight at midnight, you always have waves because you exhaust the susceptibles. And then the virus is reintroduced into another population. So you always have waves. They would just be bigger.
Yeah.
What do you think, Amy?
That's true. But so listen to this. So Osterholm was on In the Bubble. And he was talking about these waves. And he was like, I can't explain them.
Okay.
And I was like, what? And so isn't this the argument about the waves of why it's a wave every other year for 68 is because you exhausted the naive?
Yeah, exactly.
So
It's
I
exactly
don't
right.
So I never understood why for 68 it's been like this huge controversial thing that there's outbreaks every other year.
Yeah. Sydney, Australia is hurtling towards 20% fully vaccinated. 350 positives a day. So it's probably a five-fold underestimate.
But even still, that's less than like Dallas or Houston, someplace.
Sure. Of course.
Like I heard today, like had like 100,000, or maybe it was a state that had 100,000. I was like, okay, good plan.
Amy, do wild berries found on mountains contain harmful viruses and bacteria? When I told this to my dad, he laughed at me.
I'm sure they could.
Yeah, it depends if an animal has come by, but plant viruses are not going to harm you.
Right,
No.
but plant viruses aren't. But if an animal comes by, it could.
And that's probably a low frequency event. But your
Right.
dad shouldn't have laughed at you. He should
But
have
we
explained
know-
calmly.
Right. But the other thing is we know that there is harmful bacteria on a plant that can harm you because you have salmonella outbreaks all the time on cantaloupes.
And Pete took my virology course, and he says we ingest loads of plant viruses. They just pass through us. That's right.
Right.
We're good.
But she asked about bacteria too, and you do have to say that. What is the?
No, you're right. I'm sorry. I don't mean to shrug it off. Or parasites. There can be parasites, worms of various sorts. So you shouldn't just eat a berry without washing it, for sure. But
Right,
people do that all
'cause,
the
like-
time.
Well, yeah, but look at all the salmonella that's on cantaloupes
Now
or
on
lettuce.
TWIP some time ago, we talked about some parasite you could get by eating unwashed vegetables from your garden. I don't even remember what it was. So
She should have held out for Dixon.
who?
The lady who just asked the question about bacteria, and now we're talking about parasites. She should have held out about Dixon.
It would be fun to have Wouldn't it be fun to have Dixon on the live stream sometime?
It would be.
It would
But
be
Dixon
different.
all this stuff.
Dixon what?
Dixon knows all this stuff about what to eat and parasites and
I
blah
mean,
blah blah.
he wouldn't be able to do most of the virus questions, right? But when a parasite thing came up, he'd be good at it, right?
Yeah, he would.
My father has diabetes, just fought off cancer. Do you suggest a third shot for him? If so, should he get the original mRNA or the updated boosters? What do
I
you
don't
think,
know. My
Amy?
father has diabetes and fought off cancer, and I'm not getting him a third shot.
I mean, it really depends. I mean, if his response was like everyone else, then no. But, at some point in the near future, the CDC will announce boosters. I'm quite sure. And so then you can just do it. But
Well,
I don't
even
think you
if
need it.
they,
Sorry.
even once they fully are approved, they don't need to announce boosters. The doctor can just do it. It's an off prescription, whatever, off label use of the drug. Scientifically, I'm not clear that we will ever need a booster. I see no reason for it.
So should we ask Dixon if he wants to join us one Wednesday?
Sure.
Could children have had COVID up to 6 times in the last 18 months? So mild it beats current antibody tests, avoids triggering B and T cells, could be our super spreaders. 6 times? I think not. What do you think, Amy?
6 times?
6 asymptomatic, evading immune? No, I don't think
No.
so. Not likely at all. No. The Hebrew barrister, thank you so much. Stepdaughter's dad was COVID positive 2 days after she stayed with him. She had cold symptoms, fine now, antigen negative. She may have had something else. The antigen tests can be negative, but they suggest you do them multiple times, like 36 hours apart. That's why they sell them in boxes of 2 or something. So I was just reading the package insert tonight on the Binax Now. Fascinating reading. It said, if you suspect you contacted someone who's infected, you should take multiple Binax Nows within 36 hours of each other.
And how much is a Binax now?
I think a box of 2 is $20. It ain't a lick of
That's
stick.
a lot of money.
I know.
And you're lucky only one person in your family had to do it. Because if all 4 of you had to do it, that's like $100.
It's a good bottle of wine, right, Amy?
Yeah, I mean, this lovely couple has sent me 2 very good bottles of wine. Each one is about $100.
Here you go. Nick says, I like Amy, but my mom warned me about sweater girls. I love it. What's with sweater girls? I have no idea.
I don't know, but I'm a sweater girl. I'm a sweater girl all year round. And somebody recently came to my home, opened up every drawer in my credenza and said, "Oh my God, it's full of sweaters."
How many sweaters do you have? A lot?
Yeah, like 30.
Do you like them?
Some of them I like, some of them I don't like as much.
You should get rid
Some
of the
of
ones
them.
you don't want.
No!
Okay. I'm glad to see more crows again. I know Dixon isn't here, but does anyone know why seagulls seem less decimated by West Nile? No, I don't know. Dixon should be here to answer that. Do seagulls get bitten by mosquitoes?
I'm
That's
sure,
how.
but they have they either lack receptor or they have a more robust immune system. I don't know.
What's
But
the West Nile
just
receptor, Amy? Do you know offhand?
I think it's supposed to be like DC sign or something. I'm not sure. I don't think actually it's well known.
Ah, DC sign, the mannose receptor, and several glycosaminoglycans. So one of those, we think it's this, that, and the other.
But there was some question about. Oh, it was somebody was telling us about like glyc gangliosides, right? Somebody was
Yes.
saying that they thought that their virus interacted with gangliosides, even though
Yeah,
it's thought to only interact with TAM8, right? And all I can think about is how, like, when you passage like FMDV and even EV68, all of a sudden they use heparin sulfate somewhat as as a receptor, and it doesn't mean anything biologically.
glycosaminoglycans are polyomavirus receptors. Not
Yeah,
sorry, single lipids. Sorry.
single.
Single lipids. So BJR2 loves us. We formed the foundation of our accurate knowledge. Thank you. It's very kind of you.
That's very nice.
Very kind of you. All right, this one's for Amy. What's your thoughts on the optimal time gap between first and second Pfizer dose?
Well, I haven't really kept up with the literature to know what they've published. My gut feeling is 8 to 12 weeks.
Yeah. We don't follow the clinical trial timing.
No, no.
Long is good, right? Long is good, right, Amy?
Yeah,
Gives time.
long is good. I mean, we just rediscovered that long is good today or the other day. But yeah.
Immunocompromised COVID vaccine booster, is it absolutely needed? My sister's on meds to suppress her immune system. Well, they just published a study in kidney transplant patients. They got, with 3 doses, not all of them respond, even with 3. So,
Well, I
yes.
also think it depends on what kind of immune suppression you have.
Yeah, sure.
Like, are you
Absolutely.
just like some people are taking drugs that only suppress the B cells, others take drugs that suppress, something else. So I think you have to be more specific before you can, you can make a policy.
I worry about public health experts' language in regards to boost border measures, leaky borders, variant factories. Feels they're playing into the hands of certain politicians. What say you?
I don't know what leaky borders and variant factories are.
So variant factories referring to countries that have high transmission. It's just ridiculous to say that they produce the variants. You have no idea where they came from.
That's horrible. Yeah, I'm
Leaky
not.
borders, certain politicians have accused
Like the guy in Florida?
Yeah, he said Biden's allowing certain people to come in that are infested. It's just ridiculous. Totally absurd.
It's called it's called a pandemic because the entire world is affected.
Yeah, it's just messed. Jamie, the whole pandemic has been a conflict between public health and politics. So it shouldn't be. Lovely Amy, any opinion on the UK research finding cognitive deficits in those who recovered from even mild COVID?
That is true.
Yeah.
Yeah.
Not that Daniel talks about this often. At the end of each clinical report, he does a long COVID or PACS, post-acute COVID sequelae. Which one do you like better, Amy, long COVID or PACS?
Well, I can never remember what PACS would mean, so I'm not a proponent of something I can't remember what the meaning of the word is, or that abbreviation is. So that's not a good thing.
But ASPE, there are all kinds of defects, neurological defects, cognitive, motor, smell, taste. So, I mean, the extent of it is really amazing and unfortunate.
Yes.
I have heard a news blurb that trained dogs can recognize a person with COVID at 96 to 100% accuracy 48 hours before it's detectable. What could a dog be detecting? What does a virus smell like? I think that's great. I think you make organics when you're infected, certain organics, and that's what the dog is sniffing. What do you think, Amy?
Yeah, probably.
They're not smelling the virus, though. No.
No, but I think dogs can probably. I think like pheromones, like if you're pregnant or not, dogs can smell them. So I wouldn't be surprised if, due to the immune response, there was some kind of chemical that was similar.
Yeah.
Similar.
So I have to tell you,
640 people. That's a lot of people.
Hey, everybody, welcome. Let me tell you a story very briefly. There is a virus that infects aphids, and the aphid bites a plant, and the virus goes in the plant, and then the virus causes the plant to emit volatile organics that attract more aphids. Isn't that incredible?
That's
So
crazy.
maybe that's what the dog is smelling. So yes, I'm totally with that, with the dog idea. I think that's great. Hello, Vanity. Welcome back.
Hello.
Thank you again. What's the most encouraging COVID news you've heard in the last 6 months?
The vaccine that works.
Yeah, the vaccines are working more and more and more. And T cells, I think for me, the T cells are protecting us.
Yeah, well, I always thought that. I wrote a grant about it that didn't get funded.
I'm sorry.
It's okay.
You keep saying if people catch COVID and then get vaccinated, that's really good. What happens in reverse? Yeah, we had that question on Twivia yesterday. And we don't know because it hasn't been studied, but
What are you talking about? People get vaccinated and then they get infected, and if they develop disease, they get very mild disease.
No. Here's what I'm saying, Amy. The people who recover, right, and then they get a dose of mRNA. They have studied their immune response. Their antibody response is very high affinity and broad, covering many variants. So what you just said, I don't think those people have been studied. They happen for sure, but I don't think they've been studied in that great detail. At least it hasn't been published. It probably will be.
Really, Theodora? It hasn't looked at that. Are you sure?
No. I think she has not. No, but she has just put out a preprint, which we'll get her on to talk about.
She's got like 3 out.
What is your thought on Lambda?
It's a variant. Okay,
it's another variant. We don't know. It's not outcompeting Delta so far. We'll keep an eye on it. Thank you for your contribution to the incubator. When we collect nasal swab to do PCR, are we getting virus RNA from the surface or from inside cells, or both?
Both.
It's both. You get cells, you get free cells, you get free RNA. It's a mixture of things. Ooh, it's thundering out. Can boosters prevent infection? Oh, boosters. Well, so here's the story. If you get a booster and then you have a really high antibody level for, say, 2 months, yeah, in that 2 months' time, it may prevent infection, but the antibody's going to go down again, and it won't prevent infection. So, and that's normal for most vaccines. Does that work for you, Amy?
Yeah, I mean, Peter Hotez gave this whole thing about, when, you make all these antibodies and it's small leaks to the mucosal surface and then it prevents shedding. Yeah, sure. Fine.
What are your thoughts on how we vaccinated adults the first this time may have altered the course of the virus, say, compared to polio, where children were vaccinated first? I'm going to let Amy take this, and she thinks about enteroviruses a lot.
I do think about enteroviruses. So it was a childhood disease. So we did the same thing. She's missing the point. The point is we vaccinated the population that we saw got the most severe disease first. Those were the old people, and in polio, they were the young children. And then after we covered those, the high susceptibility people, we then expanded out as we produced more and more vaccine. I don't really understand.
I think it's fine. I don't think I wouldn't do it differently. Hey, Frank, welcome. Thanks for coming again. Really appreciate it.
Hello?
And then we have about 500 happy birthday Amy's.
Thank you all.
It will take me
I just
10 minutes. 10 minutes to get through. Wow, I've never seen so many. Holy cow.
Good.
You got some fans. You got some fans.
It's good.
All right, Alex, is it possible that some of these rare effects we're seeing from COVID, like long-term cognitive impairment, already exist in some everyday common cold coronas and we just never checked?
I don't know that I would have pointed out long-term cognitive impairment. I would have probably have said the loss of taste and smell were probably occurrences after the common cold coronas. But then we resolved them.
As you know, there are other post-acute infection sequelae that were known before COVID, right?
Mm-hmm. Mm-hmm.
And they may be triggered by some virus that has not been discovered. It hasn't been associated with it yet. So I think it's not something new. Although, and we have more happy birthdays now. Well, this is lovely, Amy.
It is very nice.
Very nice. It goes on for a long time.
Yeah, it's really nice. It's really sweet.
Yeah, you can't add Amy because she's not on her She has a YouTube account, but she's not on it right now because it's better not to. Otherwise, she gets echoes. Watching Michael Minna's interview with Lex Fridman, using antigen tests now and advocating for them. I think antigen tests are fine. So the idea would be to have a nice cheap one, but that never happened, right? That was the original idea. Because Amy said even $1 is too expensive.
Yeah, well, we did the math in October, right? Remember that?
Yep.
And then we did the whole thing where you said it would be by the end of the year, and my response was, in 6 weeks? Remember that? And Rich Condit was like, oh, really? The end of the year is in 6 weeks? And I was like, yeah, the end of the year is in like 6 weeks.
Look, Amy sang happy birthday to you. Another Amy sang happy birthday to Amy.
It's very nice.
The link for the town hall. Go to asv.org/education. Okay, asv.org/education. And click the button that says register, and then it'll give you a list of them. Vaccine town halls. Oh, look at Access Audio. Thank you so much. What are your thoughts on the recommendation to obtain N95s for Delta? I reckon cloth surgical would work. I don't see any evidence that Delta requires an N95. Do you, Amy?
No, but I will say that Kasi, whoever was in the bubble with Hotez and Slavit, said you should wear, you needed a better mask. And I'm not really clear why, because it doesn't really, that makes it sound like it goes flying through walls, and it doesn't go flying through walls.
It absolutely doesn't.
All right, it's 8:42, so like 3 more questions.
Okay. Vaccine's purpose is not to prevent infection?
Right, it's to prevent severe disease.
Right. Disease. All the vaccines are tested to prevent disease, not infection. The Delta variant's furin site changed from minor to RRRA is likely we will see change to a full site. What effect will this have?
So this is like McClellan's latest paper. I think it just makes it far more efficient. I think it just makes the cleavage far more efficient. I didn't read McClellan's paper yet. But
it may be good enough, John. It's hard to predict, right? What would happen and what effect it would have, if any.
Right.
Thank you. This contribution is for Amy's wine collection instead of the incubator. Okay, I have to give you $20, Amy, okay? Remind me.
Okay.
It's happy birthday.
Okay, thank you.
Martha, thank you so much for your kind contribution to the incubator.
It's lovely.
We need one more question for you, right? Should mRNA vaccine doses be spaced out longer than 3 to 4 weeks? In the UK, they did 12 weeks, and it sounds like they may be more effective.
Yeah, that's what I've always said. Doesn't matter what the vaccine is. All vaccines should be spaced out more.
Yeah, you get more time for that maturation and establishment of memory, and then you come back with a booster to really nail it down.
Yeah.
There's Marburg cases in Guinea. Oh, I didn't know. Does the virus stay in only few of the animal hosts and disappear or silently circulate? Marburg's in bats. It's actually been shown recovered infectious Marburg virus from bats. How about that? and that's the reservoir. And it periodically goes into primates, non-human primates, humans, and then stops and then goes away. And then the next cases are a new spillover.
Yep.
In what ways is SARS similar to polio, measles, or other viruses that we are well acquainted with? What comes to mind, Amy?
The RNA viruses.
Mm-hmm. They all came from non-human animals at one point.
Yeah, but most viruses, I think all human viruses came from animals.
They have caused diseases
They're acute infections.
They're acute infections. They have a defined time frame.
Yeah.
They all caused substantial disease in humans to justify making a vaccine, right? In fact, a lot of people cause disease.
Well, let's not get carried away.
With which one? Polio?
Yeah.
No,
The majority of the world was not in support of making a polio vaccine. It was solely determined by the U.S.
That's true.
The majority of the world felt that childhood diarrhea was far more important.
But, Albert Sabin used to say, we have saved hundreds of millions of children from being paralyzed with my vaccine.
Yes, that's true. However, still, billions of children have died of watery diarrhea. Rotavirus.
Yes, continue to be. Yes, exactly.
Okay. So as much as I adore Albert,
I understand your point. No, it's true.
I have a few problems with Dr. Sabin.
So
then he would have a few problems with me.
There were two. Oh boy, it would be interesting to see you and Sabin have a chat. Yeah. There are some ways they're different, of course, and that is polio and measles are eradicable because humans are the only host, and SARS-CoV-2 is not, because apparently many other animals can be infected.
Well, so far we haven't been so successful at eradicating either one of them.
Got pretty close with polio, right?
Yeah, pretty close. It's not, pretty close only counts in horseshoes.
All right, one more. Can you measure alive viral copies quantitatively, directly or indirectly?
I don't know what alive is. Is that
infectious?
Like
infectious, yeah.
Yeah, you do a plaque assay.
Yeah, plaque assay. Search my YouTube channel. You can see Amy doing a plaque assay, right?
That is true.
I bet most people on this stream don't know that. Amy is in a movie that I describe a plaque assay, and I think it's called Counting Viruses, right? It's on this channel.
Yeah,
and explain how you do it. Yeah. You measure infectious particles. You need live cells in the laboratory. It's not, and for SARS-CoV-2, it has to be done under high containment, BSL-3. So,
although Udell seems to think it's going to be BSL-2.
When did he say that?
I don't know. I just know excuse me. I think it was recently.
Amy, you're tired. Go home. I give you the night off.
Home. I have to go home. Well, I have to finish. All
Thank you for your participation.
My pleasure. I will talk to you tomorrow.
Okay. Thank you and good night.
Good night.
Okay. Let's see. Now you're stuck with me. Who knows how long. Thank you for your contribution. Really appreciate it. Is the state of the art in epitope recognition prediction such that mRNA for a custom sort of pseudo spike with a whole collection of variant epitopes could be made? Well, yes, you could. You can synthesize peptides at will. You can do thousands and thousands of all different sequences, right? But they would have to be relevant to the virus, correct? So I'm not sure what you're trying to do, but the technology, the chemistry is there. It's not a problem. But maybe you could restate the question. Yes, a certain cardiologist. He was called out. You guys all knew who I was talking about on Twiv, huh? is all foreign to me. But is Delta now confirmed to be more dangerous, transmissible, and resistant? I just have no idea. Okay, let me explain. I've always said, well, I don't see the convincing data that it's more virulent and or more dangerous, as you say. Okay, resistant, no. Vaccines prevent serious disease and death caused by Delta and all the other variants. Many people believe it is more transmissible, but many of the effects have to do with population effects, interactions of populations. So that's why we had Jeff Shaman, an epidemiologist, on TWIV yesterday. But spearfin, in my opinion, Delta is no more of a concern than any other SARS-CoV-2. If you're vaccinated and wearing masks and distancing, it's not a concern. Most of the infections that end up with severe symptoms right now, 99% of the hospitalizations are in unvaccinated people. And they're caused by Delta because Delta is predominant, outcompeted all the other viruses. How does the immune response differ from the spike produced by our own cells from a conventional vaccine, as in a single protein versus the whole virus? I'm not sure what the question is, but okay, if you take a spike vaccine, an mRNA or a vectored spike, an adenovirus vectored spike, you're just making an immune response against the spike, of course. So you make antibodies against the spike and you make T cells against the spike. And we know that antibodies against the spike will block attachment of the virus so they can inhibit infection. And we also know that there are sequences in the spike that the T cells will recognize on the surface of infected cells so they can kill the cells. They can identify a cell as infected and kill it. Now, in a virus-infected cell, or a more conventional vaccine, like, say, an inactivated virus vaccine, or even an infectious replicating virus vaccine, you would have many more viral proteins that would be targets. Now, which of them would lead to antibodies that block infection? It's not clear. Nobody's studied that very much. Maybe only the spike and a few other proteins. But most of the other viral proteins can be recognized by T cells. So a broader vaccination would give you broader T cells and perhaps more protection. And they're being looked at for next generation SARS-CoV-2 vaccines. I think we have to go beyond just making a variant mRNA. We should make a second generation vaccine that has more viral proteins in it. You could do that with an mRNA vaccine or any of the other approaches. I'm still wondering why fully vaccinated people have to wear a mask. Doesn't exposure work as a boost? Yes, it does. The CDC is thinking that vaccinated people are transmitting. That's why they want people to wear masks, to protect the unvaccinated people. Okay? I don't think there's evidence for transmission after vaccination and infection with Delta. I do not see it. But CDC wants you to mask up. That's the reason. I'm not going to say yes or no, but I don't think it's the greatest logic. But frankly, I don't think we should have stopped masking. We shouldn't have had this edict in May which said everybody unmask. Not a good idea. Actually, they said vaccinated people can unmask, unvaccinated not, but they didn't realize that everyone was going to unmask, and so now we're back to it. So we're in it to protect the unvaccinated people. That's the idea, but I don't think it's needed. Would mRNA vaccines be more effective if we used a mix? Possibly. You would have more T-cell epitopes, right? Not antibody epitopes, because I think only the spike antibodies are going to block infection, although I could be wrong. But yeah, in theory, you might have, they might be more effective. But the reason they weren't made, it was faster to make them against the single protein spike. The decision was made, at the beginning of 2020 to do that, and we're stuck with that. But now we can make new ones going forward, right? So people are going to bed. Let's see, can you explain how PCR test indicating mRNA but virus particles are not also present? So a PCR will pick up any RNA that you're looking for, right? You use primers, short DNAs, to amplify any sequence you want. So you can say, I'm going to look for spike, I'm going to look for another viral gene. It will pick up the genome as well as the messenger RNAs that are made from the genome. So it doesn't distinguish between virus, the RNA that's in a virus particle, or the mRNAs that are made in cells. And it doesn't tell you if the virus is infectious. It just tells you there's RNA there. And so if vaccinated people test PCR positive for Delta variant, doesn't mean to me that they're actually infectious. It doesn't mean that they're transmitting. It's the wrong assumption. You need to look at infectious virus if you want to make that statement. And I've said that many times before. There is a new antiviral being produced, but like the anti-influenza, you have to give very early or pre or post exposure. Yes. So probably you're thinking of molnupiravir, which is an oral drug antiviral, which is in phase 3. And you have to take it very shortly after you have symptoms or your diagnosis. Remember, the symptom onset is roughly when the RNA levels peak in an infected person. So if you wait too long, and they drop rapidly after peaking. So if you wait too long to go get your prescription for molnupiravir, it won't be effective, right? For the same reason, if you can't breathe and you go into the ICU to get high-flow oxygen, giving you an antiviral is not going to help you. Either a monoclonal or remdesivir, both antivirals, they're not going to help you because it's no longer a virus problem. It's an inflammatory problem. You have to give people anti-inflammatories, dexamethasone and tocilizumab, etc. Do you have any updates on effectiveness of convalescent plasma versus monoclonal? So Daniel Griffin on his Thursday TWIV, their clinical updates, I don't know if you know about them. They drop at midnight on Saturday. I've moved it up from 9, 10 a.m. Daniel started complaining. He said, they're coming too late. I need to take my run early Saturday. So I drop them at midnight now. So he gives updates, and he's not a big fan of convalescent serum. He's a big fan of monoclonals, and he cites study after study showing that they can be highly effective. And again, you have to give them early enough. So if you have, for example, an at-risk person, somebody over 65, someone with other diseases who might be younger, and you test positive, you give them immediately monoclonals. And that's the situation where Daniel says it can help. Not waiting until you get to the hospital. Kind of late by then. Marion, I'm vaccinated, tested positive, didn't transmit. Okay, so this paper by Andy Reid, Imperfect Vaccine, that's a paper about Marek's disease herpes virus, which is a herpes virus of chickens. And there's a vaccine for that for farmers. Farmers inoculate their chickens with the Marek disease vaccine to prevent death. It doesn't prevent infection. And the theory of Reid in that paper is that the ability of the virus to continue to transmit has led to selection of more virulent viruses. And there's some evidence for that. The problem is that those vaccines don't prevent transmission. So in your case, Marion, it could be two things going on. First of all, not everyone who's infected transmits. In fact, most of the transmissions of SARS-CoV-2 are done by 20% of infected people. So maybe you fall into that, but you're vaccinated. So what I think is happening is that the vaccination, if you're then infected, you make far less virus, infectious virus. You shed far less virus, so you don't transmit. I think you need to make a lot of virus to transmit. And if you're vaccinated, you're just not doing that. You could make a little, and maybe that's why you're PCR positive, or someone else might be. But, and so you were. You didn't make enough virus to transmit. I think that's what's going on in most cases of vaccinated people getting infected. Are children only belatedly tested for vaccine safety because we're sentimental or because they have historically been shown to be more susceptible to vaccine issues? Okay, so on Immune some time ago, Sally Permar was talking about this. She's a pediatric immunologist. And she says there's no good reason that we don't include kids earlier in clinical trials. Maybe you could say we're sentimental, yeah. But that's what she said. And I know of no reason to not include them, right? Would I recommend any of the vaccines in particular? As Daniel said, whichever one you can get. Take the vaccine you can get. They all will prevent death. We do need data on vaccinated who are hospitalized. It's being collected, for sure. Absolutely. I'm not saying we don't need data. It's being collected. I learned that the PCR test includes a human housekeeping gene. Wonder if that's how they calculate viral load. Yes, so they would use that to normalize, right, the different samples, because your nasal swab may be different amount of nucleic acid than mine. So they can use a housekeeping gene to say this is how much housekeeping gene, so you have twice as much as mine, so we're going to normalize the levels. Yes, that's how you would be able to compare CT values among different people, right? Otherwise, you wouldn't be able to do it unless you normalize the amount of RNA that's there. Okay. How hard will it be? Well, they're already being produced. So it's not trivial. You have to grow them in mammalian cells in 100,000 liter batches, and it's tricky. As Amy will tell you, if you just grow a small amount of cells, they can get contaminated, so you have to be very careful. So it's possible, and they can be purified, and it's being done at large quantities by numerous companies, Regeneron, Lilly, and others. But what's even more interesting are the nanobodies, the single-chain antibodies. They can be produced in bacteria even cheaper and easier. It's really amazing, but those aren't licensed yet. Are deer that are infected with SARS-CoV-2 a danger to humans directly? Well, I guess if you hunt and you kill an infected deer, and you dress them in the field, you could infect yourself. But I would say if you're hunting, if you're going in the woods, you should be vaccinated, because besides deer, there are all kinds of, likely a lot of other animals that are infected, like rodents of all sorts. So it should not be an issue if you're vaccinated. Why don't kids eat outdoors in school? Well, some states they could, but in other states it gets pretty chilly quickly, right, in September, October. So that's not really possible. We're getting just to the child lunch. Is it possible for any vaccine to affect ovulation? A significant other knows that blood samples show this is the case. Hello from Ireland. Hello. Well, I can't answer for all vaccines, but SARS-CoV-2 does not. And affect ovulation in what way? Inhibit it, trigger it at a wrong time? I haven't seen anything about that. Yes, everyone citing the Catter study forgets an important line that says both mRNA vaccines are highly effective in preventing hospitalization and death. Yes, when you say effective, when you're talking about a vaccine, you always have to say what you're talking about. Effective against what? All COVID, mild COVID, severe death, asymptomatic. What are you talking about? It doesn't do any good, news sources, you know who you are, to say it's X% effective unless you tell us what it is. Do you know what cells express the mRNA, therefore are targeted for attack? I answered this before. I'm sorry. We have 727 viewers. We kind of topped off at that last time. I think that's what we're going to do tonight, because it's already... When did we start? 8 o'clock. We're an hour in, and Probably not, but I don't know if we're going to get to 1,000 on the streams. I think, as I said last time, this recent interest is news driven, but our subscriber rate has been going up too. This week we had a lot more than usual, so I would see 90,000, 100, 200, 300, 400, all like every day 100, which is unusual. I welcome everyone. We're going to hit 100,000 before the end of the year. I think it's great because I want to do more teaching on this medium. I think it's cool. Is it true that PCR cannot distinguish infectious from non-infectious? That's absolutely true. I've been saying that for years, and many people are now getting it and picking it up, which is great. It never did pick up infection. It's just looking at nucleic acid. So you can imagine that there's lots of broken RNA in virus-infected cells, right? Especially in the preparation. You take a nasopharyngeal swab, you stick it in a tube, and it sits at room temperature. The RNA is degrading. As soon as the cell dies, boom, the RNA all starts to degrade. So you got bits. You have bits of RNA. And plus, virus-infected cells don't just have infectious virus particles. They have pieces of viral RNA, messenger RNAs. All of that is sampled. So if you're doing a diagnostic, you want to see if someone's positive, it's fine. But if you want to make new conclusions about transmissibility, you can't use PCR. You have to measure infectious virus. I'm sorry. It is just wrong not to do that. Thank you, Elise, for your contribution. Really appreciate it. Exact statement was, we can't reach herd immunity since the vaccine is not sterilizing. No, that's not true. You don't need most human vaccines are not sterilizing, and we get herd immunity from them. Polio vaccines are not sterilizing. They allow infection. We get herd immunity. Can I cite studies that others can share on mask effectiveness? Well, there are a bunch. I can't give them to you right now, but if you go to Daniel Griffin's show notes, microbe.tv/twiv, he has cited a few in the past months. And if you shoot me an email, I will send them to you, Vincent@microbe.tv. All right. My wife and I are vaxxed. I got COVID and rapid test positive 7 days after my wife was positive. We quarantined and wore masks around our 2-year-old and newborn, but felt pointless, I guess. I don't know, but then there's no question. I'm sorry, but masking around a young child was a good idea, just in case, right? But I don't think you are transmitting. What viral properties determine for how long viruses can last until they become non-infectious on surfaces? Well, so traditionally we have said that viruses with protein shells are the most stable, like polio viruses and common rhinovirus, common cold rhinoviruses. They don't have a membrane. And in contrast, viruses with a membrane, like influenza virus, we usually say are very labile. But the corona's kind of in between. They're pretty good for having a membrane. In fact, some coronaviruses infect the intestines of non-human animals. But what would determine how long it could last? Well, it could be the nature of the genome. In this case, RNA is more fragile than DNA, so it may not last as long. Some studies were done very early on with SARS-CoV-2 in a laboratory setting, measuring how long it lasted on different surfaces like plastic, metal, steel, copper, cardboard, and it could, depending on how much virus you put on, it could last a few days. Infectivity could last a few days. But the contribution of transmission by surfaces, originally thought to be significant, is now not thought to be a major contributor. It's mostly droplets in the air, or I should say airborne transmission. And the best virus movie is Andromeda Strain. Well, it's not a virus, it's a bacterium in that book. I have the original book right back here on my shelf. It was I bought it years ago. I loved it. It was like in college or something, high school even. But it's a bacterium. Am I not worried that any somatic cells can express? I'm not worried. Why would it matter if a few cells got attacked? You'd lose cells every day. It doesn't matter. Super chat from Kathy. Thank you. Appreciate it. Oh, let's see. Narcissistectomy. That's how you pronounce it. Okay. Narcissistectomy. Now I got it. Thank you. All right. You have to ask Amy. She's gone. What's your favorite pastime out of work hours? Well, the problem is, Mark, Amy's in the lab all the time, 7 days a week. So you might have trouble getting an answer. Did contagion end with good vaccine uptake? I'm not sure they actually tell you that. All you know in the end is it's sort of being controlled and there are fights over vaccine. People want it so badly, right? You ever see a fight over a vaccine here? No. They send them back. Vaccinated friend says everyone will eventually get infected. So might as well live up to it, go back to normal. What's wrong with that thinking? Well, what's wrong with that thinking is that you could get long COVID and you'll never be the same. Or you could die. You know, a lot of kids right now are getting very sick. Numbers are pretty scary. And a lot of kids develop inflammatory, multi-system inflammatory disease, which is quite serious. And they develop long COVID. So anyone of any age. So personally, I don't think it's worth taking the risk. And then if you get sick, let's say you're an athlete, you can never perform properly again. And many athletes have lost their abilities because of long COVID. So that's the reason you don't want to just risk it. Everyone will either get vaccinated or infected. Yeah, I agree. But we're going to have, look what's happening now. Most of the hospitalizations are unvaccinated people. It's not good. Many of them are dying. Is it possible that someone's HLA causes a complete match, thus rendering T-cell impossible? No. It's the peptide that determines whether it's recognized by the T-cell or not. And the SARS-CoV-2 peptides are not self. It's very clear. They're not self. So that's not going to happen. My 80-year neighbor's freaking out over Delta Plus. Never heard of it on TWIV. What is data-supported response for my neighbor? Well, you've never heard of it on TWIV because there's relatively little data, right? There's some epidemiological data, but the kind of data we like, experiments looking at the properties of these viruses, with the exception of neutralization with various antibodies, we haven't got. And so I personally do not think that the variants have done anything different than any SARS-CoV-2. I think in all situations we'd have outbreaks just as we've had them now. So I think there's no Delta Plus has one additional amino acid change in the spike. I don't think it's not overtaking Delta. I wouldn't worry about it. But again, we don't have a lot of data. I agree with you. But you know, the freaking out is because of the news cycle. Happy birthday, Amy. I wear a cloth over an N95 and a diving mask. Oh my gosh. Well, I think that's a bit much to wear a diving mask. Are you going out into the world with a diving mask? Really? I mean, a cloth over a KN95 is fine. 2 masks is absolutely fine. 2 different kinds of masks. That's fine, but I don't think you need to do a diving and you're fully vaccinated. No, I think the 2 masks with vaccines, are you mingling a lot? If so, that's enough anyway. Yep. Okay. Yeah, so the FDA is set to add a 3rd dose for the immunocompromised. That's based on, as I said earlier, the New England Journal and a study on three doses in immunocompromised patients. It really helps. Not everyone, but it helps. So that makes a lot of sense. Yep. Do you think there will be a day when you can affordably have access to tests for most viruses? Yeah, I think we could be there already, except that for many viruses, since there are no therapeutics, there's no justification for testing for them, right? But, we made relatively quickly tests for this virus, although it could have been a lot quicker if we had better coordination. But yes, we can. Whether we do or not depends on whether the companies can make a profit. Really, that drives it. Delta being found in St. Louis wastewater during a Delta outbreak is scary too. Why is it scary? It's another SARS-CoV-2. It's going to be there. I mean, people travel, right? There's no question about it getting everywhere. And yes, here, to quote Mark, the press look for things to spice up the variant narrative. Absolutely. And the Nature article, I saw that this week also, how it achieves its ultra-fast spread is implying that it's all because of the virus. And that's just wrong. As epidemiologist Jeff Shaman said, there are all kinds of other factors that play into the rapid spread of any virus, including people's behavior, even environmental conditions. So the implication that this is all viral is wrong. It's flat out wrong. But why am I not surprised? These are people who are just writing, and they're not working with viruses every day. Is there a danger of Marburg and Guinea for worldwide? No. Marburg does not transmit well. It never has. Neither does Ebola. So I would say relatively easy to contain it. Not a problem. So Lambda. So let's take a look at percentages of the variants. Okay, we're going to go to world in data, right? And we're going to go to world in data variants. We're going to search for that specifically. And then we can go to the lovely graph that I showed you last week. And while it's loading, I will share the screen so you can see it. There you go. Okay, so SARS-CoV-2 sequences by variant August 9th. These are the share of analyzed sequences in the last 2 weeks for each variant. So it's not everything, but it's a reflection of what's being picked up. And you can see Delta, well, these high percentages. You can click anywhere and get a breakdown for every country. So if you want to look at the U.S., hover over the U.S., and you can see Delta, 96%. We got 1% Alpha, and then Lambda 0% basically, nothing in the last 2 weeks. Gamma 1%. Okay, so in some countries, here Alpha is 1.2% in Australia. It is 10% in Slovakia. And in some countries, Gamma is picking up. So this is a change from last week, right? There's an increase in Gamma in Luxembourg, France. There's a fight between Gamma and Delta in Brazil. So Delta is pushing down Gamma a bit. And this is all to do with fitness of these viruses. It doesn't have anything to do with populations. It's just that one virus is more fit and it does better. And of course, people are contacting each other and they are transmitting it, and they're not vaccinated. So there you go. They are investigating 2 new side effects today. How are they finding side effects this late in the game? I don't know what you're talking about, Sean. They may have been things that arose before, but you have to tell me exactly what you're talking about. What use do PCR tests do if it's true they cannot distinguish? Well, it is true, Max. Do you not believe me? It is true that they don't distinguish between infectious virus and non-infectious. It is true. So what use are they? Well, as I said, if you want to know in an outbreak if someone's infected, you do a PCR test. It's likely that they are, right? And you can quantify the amount of genomes and know roughly where they are in the cycle. But if you want to make inferences about transmission, you can't use it. It's not good for saying that people transmit after vaccinated and infected, for example. You can't use it. You have to know what to use it for. You can use it for diagnostics mainly. And if you want to do some population surveys to see how many people are infected, or theoretically infected, but not for transmission. So it has its uses for sure. Are there any ways to enhance the immune response in the elderly? Well, you give them more antigen. That's why we have high-dose flu vaccine, right, which has more antigen. And the Shingrix is adjusted to people of a certain age like me. And there are other ways you could use adjuvants as well. But typically it's not done. All right, what else do we have here? We're just talking about knitting. Oh my gosh. Why can't children be vaccinated under 12? Because the vaccine hasn't been tested in them. And the FDA policy of this country is that we don't use vaccines in people in which they have not been tested, in populations that have not been tested. So, that's why. And whether you agree with it or not is irrelevant because it's the FDA's rule. Dr. R has described you as her mentor more than once. I have to admit, I just wanted to see you two react to that one. So long ago, I don't remember, but I hope you enjoyed it. Any harm getting the Novavax instead of a booster? No, I would see no harm in mix and matching, absolutely. But Novavax isn't licensed. It isn't EUA'd yet, so can't do it. Went to a translational medicine lab a few weeks ago to do shadowing. Cool stuff. Yeah, it's always good to shadow, for sure. Absolutely. You're getting your second shot Sunday. Anything to prep? Stay sober and get a good night's sleep. Nothing really specific. Pfizer seems to have worked remarkably well in the 3 counties in South Florida that constituted the vast majority of the reason we're in the big 3 infection and death rate. Politicians are the problem. Are there going to be more lockdowns? I don't know. I'm not privy to what the politicians are thinking. I don't think we need lockdowns anymore. I think we need to vaccinate people. Not a problem. UK is considering a 3rd shot for vulnerable people. Is there any way of knowing before large numbers of people start dying, you start to get, I think the idea, AFUN, is that you start to get hints whether elderly people are being hospitalized more frequently and whether more of them are dying. Now, unfortunately, that's the only way to do it. There's no other indicator because, in my opinion, serum antibodies are not a good indicator, right? Because that's not the only thing that's protecting you. The T cells are as well, and they're not assayed very often. In Iceland, 90% of the people are vaccinated, yet people are getting diagnosed at all-time highs. Some vaccinated people are requiring hospitalization. Some, exactly. Some here as well. Now, it may just be, as I said last week, this is the feature of these vaccines, that they will not prevent infection and you will get some mild illness. And maybe if we do second generations, we'll prove that. But it's better than dying, isn't it? Were the pointy sticks unnerving you? No, they weren't. I was just feeling that she's looking at her fingers and I'm talking. I guess she could listen to me. I don't know. I knew that this would provoke some questions. I think when you're in a seminar, well, look, this was a long time ago before people had laptops. Nowadays, everybody has a laptop or a phone, so I shouldn't complain. They're all doing other things on them. Knitting was probably a good thing. Get Dixon on the live stream. I'll try. Oh, boy, we are so behind on the questions. I'm sorry about that. It takes so long, but I do... I do try to answer them all. So somebody commented on the TWIV website that Dixon is useless. It's just very sad that people say that, right? He's not. I find him fascinating. I really like him. He gives you the side view. Does the spike assemble into trimer? It self-assembles, yes. The sequence dictates the assembly into a trimer. Although for some, I wouldn't be surprised if chaperones were not involved. Chaperones are proteins that can help other proteins assemble properly. Could be that those are involved as well. Well, I will try and get Dixon to join us. It is, I don't know, he can, the problem is this software I use for the live stream that I can share the chat. His slow internet will never handle it. I'll have to get him one of those cellular Wi-Fi access points. I'll pay for it, but that would help, I think. Is a molecular and cellular biology degree better than a biochem degree for study of infectious disease immunology? No, I don't think. This you're talking about a undergraduate degree, right? See, I think biochem is great because you really get to understand the building blocks of the cell. You can always pick up molecular and cellular biology later. So I would say not necessarily. I think the more chemistry you have, the better off you are. Do we have any indications yet that our intramuscular COVID vaccines can eventually induce mucosal immunity? Well, there is some IgA in a few of the studies. They have measured those, right? I think they are largely serum IgA going through the epithelial border, but they wane, right? And the question always is, so this is always weird with intramuscular vaccines of all sorts, flu vaccines, right? For respiratory infection, you would think it would be better given intranasally, but flu mist isn't much better than the intramuscular flu vaccine. So it's not just that. It could be the immunogen as well. Oh, I have to highlight this. Efficacy is the term used during clinical trials. Efficiency is what you use in the real world, right? Has Daniel treated anyone with ivermectin? I doubt it. God, this is from 2 hours ago. I don't know. I have no idea. No idea. What's the rate of polio in adults after 3 IPVs? 100%. I don't know of any polio except that caused by the vaccine. Will the rate of infection of COVID eventually be 100%? You mean the protection? I don't think so. I think some vaccines are just better at inducing high protection than others. And I think one of the features, and Amy had this idea a long time ago, is that viruses with a viremic phase do much better at inducing that kind of highly protective immunity, which of course SARS-CoV-2 doesn't do. A recent news piece stated it's possible to beat SARS-CoV-2 better than polio. I don't know what better means, right? You always have to explain your terms. We're about to eradicate polio. I don't think you can do better than that. You're never going to eradicate SARS-CoV-2. So I would say fundamentally not correct. I don't know about sweater girls. Where was your brain? I was studying. I studied all the time. I really liked studying, and I did well, and I got into a good school. And maybe that's why I do this now, right? I don't know. I don't regret it. It was fine. Is the intrinsic behavior of a virus influenced in any way by selection, or is the virus always doing the same thing and natural selection works as an independent process? Behavior of virus is always driven by selection pressure. The way a virus behaves in all ways is an outcome of selection because the virus makes lots of mutants, and then the pressures select for whatever is fit for that particular situation. And notice the word fitness, which governs everything, and which people don't like me using when I describe the fitness of variants. Of course, we use that all the time. So it's an integral part of the reproduction, right? It's totally integral. You can't take it away from how a virus evolves. You like both of our fashion choices. So yeah, I was going to wear a t-shirt today because it's really hot. I came home on the train. It was so hot. I was soaked. But then I came into the AC and I said, I'm fine. I would wear a Twiv shirt, of course. But, you know, an academic dress is not always formal, of course. And there's always a bone of contention. Let me tell you this. A lot of my female professor colleagues say they feel they have to get dressed up because if they wear shorts or a t-shirt, they'll be criticized, whereas men can wear whatever they want and they don't get criticized. So that's kind of crappy, unfortunately. But I've always dressed button-downs and khakis, always. I never wore jeans and t-shirts to the lab. Are there any viruses that can be transmitted in insect feces versus direct bites? Viruses. No, I'm not aware of any. But of course, there are parasites that are transmitted from insects. The reduvid bug, right? It bites you and then it defecates on next to the bite, and then you scratch it and you scratch the parasite into the wound. No, I'm not aware of any viruses that do that. No, it's a good question. But you know, the other issue that's related is that most viruses transmitted by mosquitoes have to actually reproduce in the mosquito. They're not just mechanically transmitted. There are a few, but most of them have to reproduce. So they have to, the mosquito, it pulls them in, they get into the gut, they enter the circulation, they reproduce in some mosquito cells, and they make it back to the salivary gland to go to the next host. Thank you for your contribution. My brother has a gene mutation that affects complex 1 of the electron transfer chain, unknown until age 5 when he had stroke. He's nervous to get vaccinated, but he doesn't know who to talk to. Input. We should send a letter to daniel@microbe.tv and we'll put it to him. Okay? Daniel@microbe.tv if you're still here, Jitsu. But I'm not aware of any, but let Daniel do that. I think asymptomatic spread from vaccinated is causing rapid spread. Take ivermectin. Oops, sorry. I shouldn't have shown that. Let's go to this one. Yes, so some sweater girls were movie stars, right? Okay, now we have a dictionary. A heterosexual woman that accompanies a gay male friend to social gatherings to maintain the appearance. I don't think that's correct. The Urban Dictionary? That's weird. That's weird. Thank you, Rob, for your contribution. Really appreciate it. Can you clarify the difference between fitness and transmissibility? The R value is not the same as transmissibility or fitness. Can you measure either of them outside of human behavior? So, Christy, you should listen to the Twiv dropping at midnight, and has to still finish editing it, so soon I will have to leave you. And I'm sorry, I'd love to stay till another hour, but I have another hour of editing to do. So yes, the fitness encompasses many properties, including transmission, but many other viral properties. So what makes a virus fit and say, which means it's really good at infecting its host, can be that it transmits well, that it evades immune responses, that it makes a lot of virus, that it makes a lot of virus quickly, that it sheds a lot. It could be all sorts of things. So transmission is one part of fitness. R is the reproductive index. It measures how quickly the virus, well, at its most essential form, how many people on average one infected person will infect, right? 2 or 3 or 8 or 10 or whatever. It is most certainly affected by multiple parameters. It has multiple components. It's got the virus itself, which is really hard to measure on its own. In fact, nobody's done that so far. Then there are human behaviors, right? Household settings, workplace settings, different kinds of settings, how many people are involved, demasking any non-pharmaceutical interventions. Immunity makes an impact as well, right? And how to distinguish is really hard. And as our epidemiologist said on TWIV, I mean, he's developed models, statistical models, to try and isolate the different factors. So he thinks he can say this part of the spread of Delta is due to the virus itself. But it's a model. It's by statistical modeling. I want experiments, which are much harder and will take another 5 years to do and may not even be definitive. So that's why he said the range for Delta R, it's no longer R naught. R naught you only do at the beginning of an outbreak. The range for Delta R, it's either called E or T. Effective R goes from like 4 to 8. He said that's because everyone has their own model and their own population, and there's a lot of variability. So it's not a constant. And we decided that when you have this table of R values for different values, to take a line from Pirates of the Caribbean, it's not a rule, it's more of a guideline, if you know what I mean. Okay, I talk enough. Thank you, Tom. Foundation of accurate knowledge. That's what I'm trying to do with this incubator. We'll see. We'll see. What are the biological principles involved in determining and setting the time between vaccine doses? All right, in this outbreak, what was the main determinant was getting a vaccine through 3 phases as quickly as possible. And so, some companies did 3 weeks, some did 4, and that's way too short. Normally, it would be months between the first and second dose because you would have the luxury of time. You don't have a pandemic, right? And so you would actually do animal studies first to say what would be a good time, 1 month, 2, 4, 8, whatever, and then pick a good one and maybe a few of those and go into people. But we couldn't do that in this case. We had to do it quickly. And so 3 to 4 weeks is way too short. You should let the first shot mature much longer, but we couldn't. Is there evidence to suggest that the variants have already waned through neutral genetic drift? Oh, I think that they have, for sure. I think there's been a combination of selection and neutral drift. And I think that also explains, influenza variation, a lot of neutral genetic drift going on as well. Tropical Philippines. Natural selection seems to have led to changes in the genome of humans that have made new coronas into common cold ones. Will we get there with evolution and vaccines? I'm not sure if it's genome changes that has done that, crazy lit as, and because it takes tens of thousands of years for a mutation to get fixed in the human genome. And so the coronas, the common cold coronas, have only been around for hundreds of years. So I think it's a matter of immunity, population immunity, and evolution of the virus. And I think that it's possible that SARS-CoV-2 will one day be the 5th common cold coronavirus, but I don't know how long that's going to be. It may take 50 years or more. Ian, thank you so much for your contribution. Really appreciate it. Incubation and libation. Vincent, who is Professor Ebright on Twitter? He says you are a cult. Oh, it's so far from the truth. Richard Ebright is a professor at Rutgers, not too far from where I am. And he is a molecular biologist. He's studied transcription in bacteria for many years. And he's an accomplished scientist in that field. But he's become a voice of opposition to certain kinds of experiments with viruses that he thinks are too dangerous. He thinks that, for example, the influenza avian H5N1 in experiments done years ago by Fouchier and Kawaoka, which studied transmission, were too dangerous. He thinks certain coronavirus experiments are too dangerous. I happen to disagree with him, but I've never spoken to him. He said I'm a cult. I'm not a cult. I just have a podcast where we talk about viruses, the kind that make you sick. I'm not a cult. Cult to me is a bad thing, right? It's like getting people to drink Kool-Aid with poison and killing themselves. That's a cult. What's the benefit of vaccines for long COVID? That's a great question. So, as you may know, 30% or so of people with long COVID say they feel better after getting vaccinated. Not everyone, not all symptoms. The question is, if you get a mild infection after you're vaccinated, can you still get long COVID? We don't know yet. Data are being collected. Have you read the study finding that Moderna vaccine efficacy correlates well with antibody? I have, and it's, first of all, it's in non-human primates, and it's efficacy. How do you define efficacy, Ty Forons? You have to define it. Remember, they're looking at infection. Infection doesn't matter because antibodies wane. You're going to get reinfected at some point because you're never going to have. They did those studies very soon after immunization of the non-human primates at a time when antibody levels are very high. Totally artificial situation because in most people, 6 months, 8 months, 10 months out after immunization, antibody levels are low. You're going to get infected. You're going to have a little bit of virus in your nasopharynx for a short time until the memory response kicks in and limits that. So the antibody response in non-human primates protects against infection when antibody levels are high. But I'm telling you, the T-cell response is what protects against disease. And really, that's what we're talking about here. Now that vaccines are easily available in the U.S., what do you think about vaccine passports? I think for travel, vaccine passports are great. I don't think they're a good idea for events here in the U.S. I think we should just get everybody vaccinated. And, for schools, I think getting vaccinated should be required throughout the country. But as you know, every state is a separate country here in the U.S., so. Are masks long-term? Oh, of course you can take them off. Most people I see have them off already. No, we're not going to wear masks forever. Absolutely not. Absolutely not. Thank you, Pam, for your contribution. Really appreciate it. Yep. You can take them. Yeah, you can take your masks off. All right, let's try and wrap up here now. I'm halfway down the questions. There's 705 people left, so some people have gone. That's fine. It's true that a mutant variant, which that's redundant. Okay, a variant is a mutant, which otherwise would be down in competition with the original form, may find advantage in an organism which blocks. Well, yeah, if the original isn't there, then there's no competition, right? Yeah, absolutely. I find it strange that some politicians don't want restrictions to curb cases, yet restrict immigration because those people might be infected. Yeah, well, there are lots of illogical constructs in politics, aren't there? Oh, dear. I had my first Moderna in March. How much would a second boost? Substantially. Go get it. Absolutely. Second boost really gives you more protection against variants. Dr. Gandhi noted that after vaccination, some people might be colonized but not infectious. Yes, absolutely. That's what we're saying here. You get infected, you're not making enough to transmit, your memory response quickly restricts it, and that is shown in the Singapore study that we talked about on TWIV. You don't transmit. I mean, yeah, you would get a boost. Absolutely. There would be little mini boosts to your immune response. And Ron Fouchier talked about that on TWIV a few weeks ago. Sorry. Please listen to Ron Fouchier's TWIV. It's really good because he talks also about influenza and vaccination and so forth. Yes. Yes. Could new vaccines and or boosters be developed that would specifically lower likelihood of long COVID? Well, we don't even know what long COVID is at this point. So I don't think we can do that until we know what it is, but I can imagine if we know the underlying pathology, but it's going to be complicated, right? And we still don't know if vaccinated people develop long COVID, so we have to find that out first. The vaccine skeptics are saying that the vaccines are driving the development of variants. Well, who cares if they were anyway? They're still protected against death and severe disease. But they're not. The variants were actually around a long time ago. That population immunity caused by infection actually selected them, and they started before much of the population had been immunized. So, wrong. I like this one comment. Do your research. Too long to explain here. Do you ever think they will find the origin? I think there's a 50-50 chance they will. Somewhere in nature. It didn't come from a lab. And earlier there were people saying that why do I say on China state TV that it didn't come from a lab? Because it didn't. It didn't come from a lab. There's zero evidence that it was in that lab in Wuhan. There's plenty of evidence that there are related viruses in nature. Easy. And I'm happy to say it as often as I'm asked to, because it's the correct answer. Well, Eddie, 71% against hospitalization. You have to look at the formula that they use to calculate it, but they take the number of cases hospitalized in the vaccine versus placebo group, and there's a formula that gives you the 71%. Doesn't mean that 29% of people are hospitalized. It's not as simple as that. More complicated than we have time to discuss here. So we're going to stop at 10. How's that? We'll compromise, and then I can go finish editing TWIV. Maybe I get to bed by midnight. Is there any way to test someone for T cells? Yeah, there's actually a test you can buy, or at least get prescribed for you to test T cells for sure. And thanks for passing 300 likes. It's wonderful. Absolutely cool. Was curious if this virus survives the digestive tract. Oh yeah, it does. We can definitely go through digestive tracts, but they're not transmitted to people that way. We have more Amy happy birthdays now. That's weird because there was a burst like an hour ago. You should 100% go on Lex Fridman's podcast. Okay, well, he didn't ask me, and he's not likely to. Why don't you everyone write into him and say to have me? You can drive it, for sure. In the plaque assay, are there virus particles released before the formation of plaques? Oh, yeah, absolutely. You know, there's a great movie of a plaque assay. If you go to my lecture number... what is it? Lecture 3, I think it's the infectious cycle, here on YouTube. It's a nice movie, and you can see, at a very early on in the infection, there are virus particles released. You can't really see the plaque. It's not big enough to see by the naked eye, but yes, absolutely. Yeah, you're supposed to do them serially because they're not so great on their own. So you do them 36 hours apart. Yeah, it's expensive for sure. Absolutely. Are there any studies on long COVID in babies? Certainly been deaths in babies and COVID in babies. And I'm not aware, but you know, part of long COVID is they're mainly symptoms, right? Which are things that you tell the doctor you have. So hard to do with a baby. Symptoms are what you can only feel. Signs are what someone could measure, right? Of course, you could measure some things that persist a long time in a baby, but mostly, you know, the neurological issues and the digestive, skin, and so forth would be hard in a baby. So I'm not aware of any. Are mice in the mouse house segregated into groups, depending on if they're going to the BSL-3 or 2? Well, they're separated already. You have to breed them in different places. You have to. So the BSL-2, we have a BSL-2 breeding area where then we infect the mice there, so we don't have to transport them anywhere. For the BSL-3, you have to breed the mice in already a BSL-3. So you could bring them in, but we don't do that in our case. DR, thank you for your contribution. Really appreciate it. How's the sun's outer atmosphere getting infected? It's not too hot, right? Amy's birthday was Monday, the 9th. I'm zipping through here just to see, if something catches my eye. Would you recommend getting one AZ and a Pfizer? Sure, I think that's fine. They have done some studies on that, and they're absolutely fine. Yeah, you could do that. Thank you, Sandy, for your contribution. Really appreciate it. On the last clinical updates, Daniel said it took 5 years to get people vaccinated. That's for polio, the U.S. Yeah. I mean, he's using that as a way of saying, we're doing pretty well with COVID, but it's a different era. I think hesitancy is really driving a lot of it, for sure, but he doesn't agree. Do we already have clinical assays that prove higher No, we don't. We don't. Transmissibility, as I said before, is a mix of virus properties and human behavior. So you have to separate them. You can't, and that's one of my main objection to the press saying the variant is more transmissible. The virus, we don't know. Statistical models suggest it is, but I want some experiments. Will the town hall be available? No, they don't record the town halls. They could. They could get me to record it, but they don't. For mutations that result in non-viable virus, could that still result in RNA picked up as positive? Yes, absolutely. It would probably be a small fraction, though, so you would never be able to see it. Really hard to do that. Kathleen, thank you so much for your contribution. Really appreciate it. Is there a correlation with EBV? A correlation of what with what, Bob? Sorry. Some people get... All right, so EBV is a latent infection. You get infected young age, and the virus stays with you your whole life, and periodically the virus starts to reproduce. And the stress of being vaccinated can cause it to begin replicating, for sure. That's been shown. Can you generate a corrected R0 which take into account relevant factors? Well, they try. There are always statistical procedures, models. They're always models, right? Which can be right and they can be wrong, and everyone's model is slightly different, so the variation. So it's not called R0 anymore. It's called RT, effective RT, or RE, effective R0, R or RT, time dependent. And there are always estimates, even as R0 is an estimate, because as you can have a highly transmissible virus, but there are no people around, it's not going to transmit, right? So you can make the estimates of the new R, but dissecting out the viral contribution from others is hard, and that's partially my objection all this time. When getting vaccinated, should a person make sure that the needle is aspirated? Well, Daniel has said it doesn't matter. Some countries think it matters. I've seen many people not have it aspirated, and they're fine. So Daniel says no. So I'll listen to Daniel because I talk to him every week. Thank you, Neil, for your contribution. All right. 5 minutes. Let's see. Can you explain what an epitope is? Epitope is a part of a protein that is bound by an antibody. So it's a short sequence of amino acids, 8 to 12 amino acids. And the antibody molecule that your body makes attaches to that. And sometimes a one amino acid change in that epitope can cause the antibody not to bind so well. So Spike has antibody epitopes all over the place. Then there are also T-cell epitopes. T-cells recognize infected cells because they have on their surfaces small peptides that the cell puts there. The T-cell can recognize and see if it's foreign or not. And if it's foreign, they can kill the cell. How can a PCR ever give a false positive? Oh, it can have off-target binding. It can have error, a user error. It happens all the time. Fauci's talking about oral antivirals, molnupiravir. Look it up, MOLNUPIRVIR. Oral antiviral phase 3, looking good. It is really looking good. Merkel is the number one perpetrator of anti-vax information, by the way. He makes a lot of money doing that. It's really unfortunate. I wear a mask every time I go in public. I have grandkids too young. I do not mask with them. Am I doing this wrong? Should I be masking? I think, are you vaccinated? You probably are. I think you're fine. If you're masking religiously outside, you are okay. How would you best respond to someone who's using the VAERS database as evidence for anti-vax sentiments? Well, I would say the VAERS captures everything that happens in a person's life around the time of vaccination. So if they break their leg, they report it. If they die, they have a heart attack, they're in a car accident, those are all reported. So it overinflates the apparent issues, right? But then they go through them one by one and they say, no, this is not having anything to do with the vaccine. Hope that's enough. That's what I would do. And you want to give them not too much because then they get turned off. All right. Thank you, Overjoyed, for your contribution. If you're vaccinated and catch COVID, should I still get monoclonals? I have severe asthma. So Daniel Griffin says if you are in that category of people that could have a severe issue, yes, you should get monoclonals for sure. Okay, let's go through the super chats. Richard, thank you so much for your contribution. And absolutely, folks are answering questions for Daniel, with Daniel, and that's great. Because he's the clinician. I'm just a virologist, right? But I think the team is pretty cool, right? That we cover all the angles. Thank you, Ditching the Grind, for your contribution. And those of you who didn't get your questions answered, please come back next week with them. And this is all recorded, of course, if you missed some of it. But I would love for you to come back and ask again. Thank you, Les, for your contribution. The Incubator, folks, is the name of our new studio. Thank you, Pete. Sent directly to Microbe TV. Really appreciate it. Look at that, 350 likes. Maybe next time we could get 400. Thank you, Vanity. If I write you a virus parody, will you sing it? Sure. Absolutely. Would love to. Thank you, Ralph, for your contribution. If you're not a cult, well, then why are your podcasts and live streams so addictive? Well, I thank you for thinking they're addictive. I appreciate that. It's funny that just people talking would be addictive. It's just not the word I would use, right? So there are very popular people on YouTube, like Marcus Brownlee, right? Who I really like. I think he does a good job. He has millions of followers, but they don't call him a cult. They just like him, right? They like him, and they like his style, and they like what he gives them. So I guess people like what I give them, what we give them, right? Cult. That's the funniest thing anyone has ever said. That's really funny. I have to remember that. Oh my. Thank you, John, for your contribution. Tomorrow I'm going back to the incubator, and you love it live. I want to do more live. I think, the recorded conversations is great amongst us because, it's different with a live stream, but I want to do more live, as I said, and that's one of the advantages of having a studio where I can go and do that all the time. All right. I think that'll do it. We're at 10 p.m. Thanks for coming, everyone. I really appreciate your being here and listening and chatting amongst yourselves. And do come back next week. And until then, I want you to stay safe, right? Thanks, everyone. Good night.